Finally, we wanted to consider the nucleus to cytoplasm size. The nucleus is going to be, as we'll learn, and probably most of you know this already, the nucleus is going to be the control center for the cell. If we had cells of various sizes, say a relatively large cell and a relatively small cell, typically we would expect to find just one nucleus, and I've got two. All right, let's see if I can join these together. Okay, we've got one nucleus in there. If that's supposed to be the control center, it's going to be able to control the cell without any problem at all. On the other hand, if we have a very large cell and we now have a nucleus of comparable size in there, we have a much larger area that'll have to be controlled, and we're going to start running into communication problems. How are we going to maintain control, or how can we still be the brain if the body we're trying to run is too big for us to communicate effectively? So the number two reason basically is how are you going to maintain control if the brain doesn't fit the body, essentially. I would, however, like to work through something with number one in terms of cell volume to surface area. And if we can come in on the overhead camera, just to illustrate a couple of points here with this one, I'm going to try to draw a cube as best I can. Okay, if you can envision a three-dimensional object here, a cube, and this is going to be our cell. Now, a cube has 6 sides. So the surface area of the cube is going to be equal, if we wanted to do a mathematical formula here, it's going to be equal to the length times the width times s, the number of sides. Volume of the cube is going to be relatively easy to figure out. Mathematically, that is length times width times height. Length times width times height. Surface area is length times width times the number of sides that we have here. So what I would like to do is to change the side, or how big our side is on this cube, and then take a look at what's the surface area, what's the volume, and then, very importantly, what's the surface area volume ratio, or volume surface area ratio. You can look at this in either way. I think in the still store we had it as volume surface area. So we're going to take this cube and look at it on a side. Okay, if we start off with our cube so that the side is equal to 1 unit, whatever unit we want to make here. It doesn't matter right now, we're just going to say it's 1 unit. Okay, if that's the case, then our surface area is then going to be equal to length, which is 1, times width, put in what I want, but which is 1, times the number of sides, which is 6. So that's going to be equal to 6 units squared, basically. Volume is going to be equal to 1 times 1 times 1, length times width times height, and that's going to then be equal to 1 cubic unit. If we then take a look at the surface area volume ratio, that's going to be 6 over 1, or our ratio is 6. We have 6 times as much surface area as we have volume. Okay, so that's if we have our side equal to 1 unit. Now, let's take a look at what happens to the cube if now, instead of having our side being equal to 1 unit, we make it equal to 2 units. We're going to make it twice as big as what we had before. So we slide this out of the way. And now if we have the side equal to 2 units, surface area is then going to become 2 times 2, length times width, times the number of sides, 6. That'll then become 24 square units. Volume is going to be equal to 2 times 2, length times width times height, or 2 cubed, is equal to 8 units. So now if we take a look at surface area to volume ratio, we now have 24 over 8, or the ratio now becomes 3. If you compare that to the ratio that we had a moment ago, where we had the unit being equal to 1, instead of having a surface area volume ratio of 6, we now, in doubling the side, have halved the ratio. So we now have half as much surface area to work with to feed our particular volume or guts of the cell. The ratio is going down as the sides are getting bigger. In fact, let me take this maybe just one step further as an illustration. Let's now make the number of the sides, or the units of the side, equal to 5 units. We're going to make it 5 times as big as what we started off with. When that's the case, surface area is going to be equal to 5 times 5, length times width, times the number of sides, which is 6. That's going to be 150 square units. Volume is going to be equal to 5 times 5 times 5, length times width times height, 125 cubic units. Surface area divided by volume now comes out to be 1.2. Our ratio is going down smaller and smaller and smaller and smaller. Of the two materials, surface area and volume, the part that we really need to be considered with in terms of keeping a cell alive and functioning is surface area. So what we want to do is to have a size that maximizes this ratio, or in other words, keeps surface area relatively large compared to the volume, so that we can maintain the diffusion processes and keep things going inside the cell. In that particular situation, our best ratio then, or our best way of doing it, is then have small cells. Now, we have one other way we can take a look at this. If we had our cube, and I'll see if I can find our cube diagram here. If we had our cube, what we could do is subdivide and make little blocks out of each of these things. If we now make little blocks out of it, then each block individually is going to have a very favorable surface area to volume ratio. So one of the reasons why cells seem to be small is because it's the best way of maximizing the surface area volume ratio in a favorable proportion. By having a small cell, you have a proportionally higher surface area to volume ratio, and that's extremely necessary for diffusing materials in and out of the cell. Okay? If you have some questions on that, if you weren't quite following my math, you might need just to sit down and play with it a little bit to convince yourself that surface area is going to be length times width times the number of sides. And with the cube, we have 6 sides. It's a very easy geometrical form to work with. Volume in this case is equal to length times width times height. Plug in some numbers for units, just as I did here, and then take a look at what happens to the surface area volume ratio and convince yourself that this is going to be necessary to maximize the largest number that you can get here, which will be favorable for life processes like diffusion and osmosis. Okay, let's leave that particular point behind. Get some of this stuff cleared out of my way here. And let's go back to the still stores at this case. We're going to mention a basic cell organization and start discussing various materials in turn. If we take a look at a basic cell and we start breaking it up, one of the 4 main components that we will find will be a membrane. We've already talked a great deal about membranes and how membranes are put together. We spent some time on this, so there shouldn't be any real surprises there. Historically speaking, one of the other chief areas that we would mention in the cell is going to be that of the nucleus. In fact, a very historical way of looking at it is to divide the cell into the area that is nucleus, such as here, and the area that is non-nucleus, all the other material that's out there, and that's going to be referred to as the cytoplasm. So if my blue line on the outside represents the membrane, the blue circle on the inside representing the nucleus, the cytoplasm, the third component here, is going to be all of this material here, everything that's in between the nucleus and the membrane. Organelles, on the other hand, are relatively recent structures, and they're going to be scattered either in the cytoplasm, or actually speaking, the nucleus is very much like an organelle. It can be considered an organelle. So organelles are going to be some of the more specific cellular structures that we're going to run into here. Okay, let's go to the next still. The membrane is basically going to separate the cell from the environment. That's what we've talked about for membrane purposes all the way around the board. No real problem on that one. It's going to be serving as a boundary layer for regulating the movement of materials either in or out of the cell. So if you don't quite get all this copied down, folks, there's nothing really new here. This is the stuff we've been talking about previously with membranes. Every time we have a membrane, it's going to be doing these main components. It's going to be separating the cell from the environment, and it's going to be regulating the movement of materials into the cell or out of the cell. It might be one of the passive movements that we talked about with diffusion and osmosis, or one of the active transport mechanisms that we talked about on Monday with pinocytosis, exocytosis, and so forth. Okay, let's go to the next still, please. Cytoplasm, as I tried to indicate in my kind of drawings with the light pencil here. Actually, this is my light pencil. We use a fingertip here instead of a pen. Cytoplasm is going to be the cell substance between the cell membrane and the nucleus. So if we have our cell and we put a nucleus in it, cytoplasm is going to be all of the gunk in between the membrane and the nucleus. This is sometimes called the fluid component of the cell, and without use of special microscopic techniques, it doesn't appear to have a lot of structure or organization to it. In fact, there's a lot of things that are there in the cytoplasm that weren't recognized until we had the better microscopes, transmission electron microscopes, and so forth, and that's part of where we're going to focus things. So cytoplasm, more of an historical term. If you break it down, cyto we've seen before, prefix meaning cell, plasm referring to substance or body, so cell substance basically is what the term cytoplasm refers to. Okay, let's bring up the next still. Organelle, that third component, the term literally means small organ. If you put that L suffix on the end of it, it's a diminutive, so it's meaning small organ. Basically, this is a body within a cell that's doing some kind of specialized function. We're going to get a whole bunch of things that are going to fall into the terms or under the classification types as organelles. The nucleus, as we mentioned, and lots of other things that you're already familiar with, like the ER, mitochondria, chloroplast, all those types of things are going to fall into this. Main importance of the organelle is this one right here. They're important because they allow us to compartmentalize and organize the guts of a cell. It's kind of like if you visualize a large warehouse. Yeah, it's good for doing some things, but if you then put in floors, break off offices and rooms and so forth, if you compartmentalize that large space, now you have the ability to get a lot of different functions all done simultaneously without one interfering into the other one. So the organelles are going to be extremely important in this compartmentalization role and the organization of cells. We're going to be spending some time looking about how each organelle is put together, what their particular functions are, and so forth. Okay, let's go on to the next still, and we're ready to start doing a discussion here. Now, folks, in our discussion with these substructures, there's two things I want you to function on. I want you to know function, what does it do, and structure. How is it put together? Are there any unique components about how it's put together? So structure and function, that's the things that you definitely want to be able to give me. A third component that we'll mention from time to time, and that'll kind of develop as we go, as which types of cells have this structure. Is it a plant structure? Is it an animal structure? Is it something that's found in both? Now, nucleus, the obvious one to begin with. This is typically the most conspicuous organelle. When you take a look at a cell, this is the one that everybody can usually find right away. Sometimes they're pretty translucent, but typically this is one you can find reasonably fast. They are usually spherical. I don't know if you've ever noticed that before, but the nuclei are typically spherical, but they don't have to be. They can be lobed, that is, they have little lumps on them, or they can be somewhat irregular in shape. Now, this business, folks, is an FYI piece. I will not test on this, but you might keep this kind of an idea in terms of approximate size. Usually, they're around 5, and this little funny U with the stem on it is that symbol for micrometers, a 1/1,000,000th of a meter. Nuclei usually range or average in size about 5 micrometers in diameter. That'll give you an approximation. That's pretty good size. Nuclei are one of the largest structures or one of the larger organelles. Usually, but not always, we tend to find one of these per cell. We do have some cells that are enucleated or anucleated, don't have a nucleus, and we do have some types of organisms or cells that are multinucleated. Those are a little bit unusual, but general rule of thumb, 1 per cell, 1 to a customer. Okay, next still, please. Okay, a couple things about the structure of the nucleus, and this is where we want to get down into some details and so forth. There is an envelope, a membrane that surrounds the nucleus. This is what's referred to as the nuclear envelope. Nuclear membrane. We've got a couple of more specific things to say about it in a moment. The nuclear membrane or nuclear envelope is not a solid structure in that there are pores embedded in it. So we're going to take a look at the holes, if you like, within the nucleus. There are also typically 1 or more nucleolus or nucleoli, if you want to use the correct plural on that one, a nucleolus. We'll have some more information on that one in a moment. And then chromatin, if you remember this chrome prefix, it refers to colored. Aton referring to threads, so literally colored threads. This is going to be the genetic information, the DNA and so forth that's found inside the nucleus. But in its first visual appearance with the early microscopists, with the use of stains, it tended to look like kind of a loose colored, springy, thready type of material, and the term chromatin was described or named to describe that condition or describe that information. Okay, let's go to the next still. Now a little bit about the nuclear membrane. The nucleus is one of only 3 organelles that we'll run into, which is double membraned. That is, it's going to have 2 layers of membrane, a double sandwich if you like. That's something that will become apparent with some of the electron micrographs that we'll see from time to time. But there's a double membrane there. That is unique. That is a characteristic of the nucleus. Typically, the inner surface of the nucleus is coated with a structural protein that will help it maintain its shape. You might, in some of your readings, run into some references on what's referred to as the crystalline protein. Basically, this is a gelatinous material that helps keep the protein or keeps the nucleus in shape. It's possible, for example, for you to go in with a microscopic hook, grab the nucleus, yank it out of the cell, and then bounce it around on the microscope slide like you would dribble a basketball. And the main reason the whole thing doesn't crush and collapse on you is because of this protein that lines the inner surface. There is a space between the 2 membranes. This is kind of an FYI thing. I don't think we'd ever test on this. It's about 20 to 40 nanometers. And this perinuclear space, peri meaning between, so the between nuclear membranes, there's a space there. So the membranes are not exactly solid all the way through. Okay, hopefully we've had that one up long enough. Let's go to the next still. Okay, the nuclear pores. These are actual holes in the membranes, and I think you've got some nice diagrams of these in the Swanson book that you can take a look at. One nice thing about the pores, they run through both layers of the membrane. So this isn't a hole in just one of the layers. The hole runs all the way through. It's a complete tunnel or channel, if you like, a porthole that goes all the way through both layers of the membrane. There is a protein structure that regulates and makes the hole there. A little side note, it's made out of an octet. There are 8 protein units that make the shape. And we can close the hole. We can put a plug in there and close the whole thing off. It's thought that the nuclear pores help to regulate movement of materials in and out of the nucleus, that these are involved with moving things in the nucleus, out of the nucleus, and so forth. Okay, so kind of summary again. I'm hoping we're putting enough time in here for you to get this down. We've got nuclear pores, which are going to be holes in the membrane. They do run through both layers. They're made up of protein. This just isn't a gap. This is actually a structure that's built into it. It's kind of like a window frame, if you like, in the side of your house. And it's thought to regulate movement of materials in and out of the nucleus, so the nucleus can communicate with the rest of the cytoplasm, the rest of the cell. Okay, let's forward to the next still. Now, the nucleolus, one of the other structures that we mentioned that's typically found in a nucleus. Usually, although not always, This shows up as a dark area inside of the nucleus. In some of the different light formations that we can use, this may show up to be a relatively translucent area, or at least some area that's a little bit denser than what the rest of the nucleus is. We may find 0 of these. There may be 0 nucleoli in the nucleus, or we may have multitudes of these. And they also will disappear depending on what the cell's doing. If the cell's getting ready to divide, for example, the nucleoli will disappear. We have linked these with the function or with the synthesis of a cellular structure, which we'll discuss in a moment. That structure referred to as the ribosomes. So they are involved with the synthesis or involved with the structure of the ribosomes. Okay, while you're getting that, let me pop up a laser here. Having just a little bit of coordination trouble, I've got things on 2 different lasers and how to bounce back and forth on those. Okay, let's go ahead and bring that laser up on screen and we'll get ready to play this one. This is a time-lapse movement of allium cepa. This is a material we'll be looking at probably in lab relatively soon. This is an onion, and what you're looking at, folks, just to kind of set you up, is that you're looking at a cell which is actually larger than the whole frame, and what you are interested in is this structure that I've encircled right here in the middle. This is going to be your nucleus. In fact, if you look very, very closely, you might be able to pick out a nucleolus. All the other material that's around here surrounding it is going to be cytoplasm, cell wall, cell membrane. We can see at the top, maybe a little bit of it down here at the bottom. But let's go ahead and play this one and see what they have to tell us about the nucleus.
The cell nucleus, in this case from an epidermal cell of the cooking onion allium cepa, is surrounded by rapidly streaming protoplasm. The nucleus contains most of the genetic information in the cell. In the 2 nucleoli, ribosomal subunits are produced.
Okay, nucleoli or nucleoli, there were 2 of them in this one. I don't know if you picked it out. I could see the lower one quite easily. The upper one is not real observant to me here in the studio. You might have noticed the movement in these channels or these threads moving away from the nucleus. This is a phenomenon that I hope we'll be able to see in lab. This is what we call cytoplasmic streaming, and this is where we see the cytoplasm, which is fairly dense, moving in relatively thin channels because there's something else occupying the space that it has to go around, and that's another structure that we'll be talking about. Okay, while we're on this one, let me bring up another one. Make sure I hit the right buttons here. Okay, this one is a little bit different. This one is an organism known as acetabularia, which interestingly enough is usually multinucleated, but they're going to show you an image from a single nucleus. The whole clear area is the nucleus, which should tell you something about what the structure is, the dark structure inside. So let's go ahead and play this one then.
In the green alga acetabularia, the nucleus from the zygote contains a very large and dense nucleolus. During subsequent development, the nucleolus grows and divides into subunits. A mature cell has a giant nucleus with a diameter of 150 micrometers. A large number of nucleoli are recognizable within the nucleus.
Now, if you didn't catch that, folks, this organism has a nucleus that has a size of 150. Can't get this thing to draw where I want it to. 150 micrometers, okay? The FYI piece that we gave you earlier is that typically the nucleus is 5 micrometers in size. So this thing has a nucleus which is over 100, well, not quite 100, over whatever the figure is, 20 times larger, over 20 times larger than what we normally get. So the whole structure you see on the screen here is one giant nucleus, and each of these little spheres, which we see right now, which almost look to be nuclei, are the nucleolus, or the nucleoli that are inside of there. This particular algae has a very interesting reproductive cycle, what would happen, and we might go into this later on, is that this nucleus is going to break up into pieces, and then each one of these pieces is going to go off and form a different gamete or a different sex cell. So that's why we have this particularly unusual structure showing up here as far as what's going on. Okay, let's come back in and go on to the next still store. Leave the laser alone for a little bit. Okay, the chromatin that we were talking about, the name literally does mean colored threads, and basically this is your DNA and the associated proteins that keep it together or keep it organized, kind of set up in a loose format, if you like, having it set up so it can do various things. Okay. Trying to find something here. Alrighty, got what I needed. Okay, let's go on to the next still. Trying to do two things at once here. Nucleus function, as it's been mentioned before, and this is probably nothing new to you, this is going to be the control center of the cell. Often this is said to be the command center, the brain of the cell, you know, quote unquote. This is the area that's going to be running the cell, helping directing the cell, letting it know what it's supposed to be doing. So with the structure of the nucleus, we've had several different things with the membrane, with the nuclear pores, with the nucleoli, and now the chromatin, but the overall function of the nucleus to serve as the control center of the cell. Okay, let's go on to the next still. We're going to bring up another structure here. By all rights, probably shouldn't be considered an organelle, but it is going to be a very common cell structure and one that is found in both prokaryotic cells and in eukaryotic cells. You might remember this one from the diagram. So ribosomes are found essentially in all cell types, prokaryotic and eukaryotic, and they are also found inside some of the other organelles. When this happens, we'll be sure to mention this or bring this up. These are going to be found in some of the other organelles. Relatively speaking, ribosomes are very, very tiny. They average on the size of 20 nanometers by 30 nanometers. This is a whole scale, whole what do I want to say here? Magnitude, that's what I'm after. A whole magnitude scale different in size than what we had at the nucleus. Usually there's quite a few of these per cell. You may end up having quite a couple 100,000 to a few million. They're very, very numerous in cells. So a relatively common structure found all over the place with cells. Okay? If we take a look over at the laser for a moment, we'll give you a sneak peek at these things. There's not a lot to see with them. If you take a look at them, and this is under a TEM and extremely high magnification, these little itty bitty black dots that you see on the screen are your ribosomes. And you can pick out that there's quite a few of them. They tend to be arranged. You might see that some of them are in lines or in channels running through here. There's some real nice lines of them up here in this area and so forth. There's a lot of these, whole bunches of these in the cells, but they're very, very tiny, and you need the very high magnifications in order to pick them up very well at all. Okay, let's go back to the stills. Keep Rich jumping around here in this new studio that he's not too familiar with. Structure on these. Ribosomes are typically made out of two subunits. And the subunits are made out of the chemicals or the macromolecules of protein and a form or type of ribonucleic acid. Type of nucleic acid known as little r RNA or ribosomal RNA. Had this from the macromolecule worksheet. One thing about the ribosomes, and this is where you get some splitting about them considered as an organelle, there is no membrane that surrounds the surface of the ribosome. And this is where some individuals would split off whether these are going to be considered as an organelle or not. Typically we think of an organelle as being a membrane-bounded structure, and ribosomes do not have a membrane around them. They're just a chunk of protein with some pieces of ribosomal RNA. Okay, let's forward to the next still. And I tell you what, while you're copying that down, let me flip over to the laser player and change laser disc. So Rich, you might just want to take me off screen while I move off camera here. As I said, one of the trouble, we've got two lasers with two different things on them, and I'm having to flip back and forth between them. There are two subunits with the ribosome. They are very originally named as large and small. And we have a few factors up there concerning both of them. Large subunit has roughly about 45 different proteins that make it up. And there's also 3 ribosomal RNA molecules that make it up. Small subunit, not surprisingly, is smaller. It has only about half the protein number, only about 23 proteins in there, and only 1 ribosomal, oops, I deleted myself here, 1 ribosomal RNA molecule. Okay, let's take a look over at the laser. I got the new one installed here, and we'll take a couple looks at some things. This cluster of materials that you see here and so forth are clusters of ribosomes at extremely high magnification. You may just about be able to make out in some cases that there's a dumbbell shape or a snowman shape to them, where you have one large unit and one small unit kind of hooked together. You may have to kind of stretch your imagination a little bit on these, but there are 2 subunits on there, 2 subunits in the ribosome. Also at this magnification, we've gone up just a little bit closer in. Each one of these little boulder-like structures is a ribosome. And I don't know that you can really see the 2-piece size or 2-piece construction to them very well with this magnification, but they do break apart into the 2 subunits. Okay, we do have a model here on the laser that'll show you a little bit about the ribosome. The molecule, or this tape that you see running down through here in the middle, is another RNA molecule, messenger RNA, and we'll talk more about it later on. But the yellow piece of the model is one of the subunits, and the red piece of the model is the second subunit. It is possible for these two to come apart. In fact, we can take them apart, put them back together, and so forth, and the messenger RNA tape does run down in between them. So they're not perfectly spherical. You can see these little lumps and bumps coming off of them. Might have picked that up from the EM pictures as well. And if we rotate this over, here's the large subunit in the red and the small subunit up here in the yellow, and then our messenger RNA tape running down through the 2 of them on the one side. So our 2 subunits on the ribosomes. Okay, let's go back to the laser, or excuse me, the still stores. Make sure I know where I'm at here. Okay, function of the ribosome. So we've talked about their structure, how they're put together. Function is that they are going to serve as the site for protein synthesis. We've talked about linking amino acids together, getting the dipeptide bonds to form in order to link up the proteins, making our primary protein structure. This is where we actually do it at. This is going to be the function of the ribosomes, is protein synthesis. We can also take a look at the ribosomes in terms of their location, and that's what is referred to as types of ribosomes. We can have some ribosomes that are floating free in the cytoplasm, just kind of hanging around, doing nothing, hanging out there in the cytoplasm, waiting for something to happen. Or, very commonly, they may be bound to the membranes. And it's when they're bound to the membranes that you end up seeing these dots, like we saw in some of the earlier micrographs, where you see these rows of ribosomes all lined up on top of each other. That's typical of the membrane bound. Ones that are free in the cytoplasms tend to be scattered around all over the place without having a lot of structure to them and so forth. Now, once I have this laser flipped over here, let me bring up and go back to something. Okay, hopefully we've had enough time to copy that over. Okay, let's bring up the laser. This, however, folks, is not a giant ribosome, although there are some ribosomes in this little area out here. What I've done is backed up a little bit to pick up the still stores off, or the laser images off this other disc. What you're looking at should have been almost immediately recognizable to you as a very high close-up of a nucleus. Chromatin is going to be some of this darker, grainy material that we see through here, and little bits and pieces of things, never really a clear shape or form to it. The darker staining structure that we picked up is going to be the nucleolus. Nuclear membrane is evidenced through here, but we're not going to be able to see real clearly the double layer of the membrane or the nuclear pores in this particular magnification or shot. Okay, and we mentioned about the organism in the film acetabularia. One of the things that we do know is that the nucleus does control function within the organism, and this is some of the experimental proof of this. So again, I'm kind of backing up a little bit on some of the material. What we do know is that with acetabularia, you have a base with the nucleus down here at the bottom. And I don't know if your screen's large enough to see that well, but we have a nucleus down here at the bottom. If we sever this cap structure and this stalk, take all this off so that we're left with just a cell, the cell will regenerate a new cap and new stalk. But the shape of the cap is controlled by what's down here in terms of the nucleus. And the way we know that is that it's possible to find two different species of acetabularia which differ in the shape of their cap. If we do some transplant situations where, let's say, we transplant the nucleus around onto the cap, or the stalk rather. So we're going to bring this base with this stalk and we put them together. The cap that we get back is the cap from the nucleus one, not from the stalk. That tends to show us that the control mechanism for what grows the cap is not here in the stalk. The information for growing the cap is what's down here in the nucleus. And there's been a whole series of really nice classical experiments on this which help to illustrate that it's going to be the nucleus that's involved with controlling things. Okay, Rich, I've lost track of my time. How long have we been going on the tape? We've been 45 minutes? Okay, all right, let's go ahead then on to the next still store. I won't get all the way through it, but we've got enough time to introduce the next topic. Okay, our third structure within the cell, if you like. We've had the nucleus, we've had the ribosomes. Structure number 3 with a horribly long name, endoplasmic reticulum. Folks, nobody talks about this in its full name. They always refer to it as the ER for short. Actually, if you break down the word endoplasmic reticulum, endo means inside, plasmic referring to substance or body, so inside the body, and reticulum is a nice fancy name for network or lattice-like type of material. So we're talking about inside the body network, basically, is what the name endoplasmic reticulum refers to. It is made up of membrane, and it is a huge component of membrane. It makes up to as much as half of the total membrane that's inside of a cell. In fact, this thing is pretty much nothing but membrane, but there's a very huge quantity of it, depending on what cell you're taking a look at. So there's absolutely huge quantities of ER out here. The ER membrane often is continuous with the nuclear envelope. Now, what do we mean by continuous with it? Well, if we have our nucleus, our nuclear envelope, and I'm trying to draw something like here, the ER, which normally looks as a lot of parallel lines, very frequently runs right up and joins to the nuclear envelope. We often have the membranes apparently running from one right into the other one without being any conspicuous breaks on this. That's what we mean by continuous. Okay, if we forward to the next still, hopefully that's been up there long enough. In terms of structure of the ER, it's basically a bunch of folded membranes. So when you take a look at this with the TEM and so forth, you very frequently see a series of parallel lines, and this is the membrane lined up on top of itself. So if you run into a bunch of parallel line type things and you're looking at it with the TEM, basically ER, that's what you're going to have. Now the membrane does have the capacity to make little sacs, so we can have some flattened areas or sacs, and these are sometimes referred to as cisternae. It's another one of those vocabulary words we can throw at you. All we're really referring to is that the membrane can make little pockets on it. Those pockets are called cisternae. And the membrane is also bifacial. Now we ran into that term when we talked about the cell membrane. Bifacial meaning that it has a very distinct outer layer because of markers like carbohydrates and things like that, as opposed to the inner layer, the O and I out here. So we've got a very distinct inner and outer layer on the ER. It's not just a random set of membranes that you can run either direction, that type of thing. Okay, I think what we'll do is call it at that particular place. Rich, why don't you, well, okay, we'll have that maybe as the starting point if we need to on Friday to finish up on things. What we're planning on doing for tomorrow is bringing in some live material that we can take a look at with the light microscope and look for some of the materials that we have been talking about. I'm looking predominantly to look at cheek cells so that we can do a cheek cell scraping and stain it with methylene blue and look at some human cells. Hopefully that'll bring up a nucleus. We should be able hopefully to pick up a nucleus maybe in some other plant cells like onion, and then we'll just see how far we get. So we should be doing some lab work on air tomorrow, taking a look with the microscope at some of the materials that we have here, and I may pop in a few extra things like the staminal hairs of Tratus scantua, a couple other things maybe to take a look at. But Greg, if you're setting up for preparation, if you can do the cheek cell toothpick scraping, methylene blue for staining, and then probably white onion epidermal peels, and maybe Eladia. I'll see whether I can get some at the local pet store here in the next day or so, have that ready for us on Thursday. We should get just a few things to look at, give you a chance to look at some stuff, and then we'll put these up on the microscope on camera as well and see what we can take a look at. Academy students, hopefully you've paid pretty good attention as to what's going on. If you have some of the reading materials, you can turn it in with the attendance sheet to the person who's here with you today. They'll pass that on to me. Otherwise, we will see you in lab Thursday evening, and we'll start doing some microscopic work with you folks. Okay, again, if you had any questions, things that came up during the broadcast that we might need to elucidate a little bit further, hopefully you've dropped that or jotted that down. We should have some time either on Thursday's lab broadcast or on Friday where we can backtrack a little bit, take care of those types of loose ends. Other than that, folks, thank you for being patient with me with this pre-tape broadcast, and we'll talk to you on Thursday.